Hello friends, today we will learn about translation and regulation. Translation and protein synthesis is a process during which the genetic information is translated following the dictation of the genetic code into the sequence of amino acids in a polypeptide gene product requiring the functions of a large number of macromolecules. In simple words, it is a process in living cells in which the genetic information encoded in messenger RNA, that is mRNA, in the form of a sequence of nucleotide triplets, that is codons, is translated into a sequence of amino acids in a polypeptide chain during protein synthesis. Now come to let's know about the difference between prokaryotic translation and eukaryotic translation. Prokaryotic translation means it occurs on 70S ribosomes, but eukaryotic translation occurs in 80S ribosome. Prokaryotic translation is a continuous process as both transcription and translation occur in cytoplasm. But in eukaryotic translation, it is a discontinuous process as transcription occurs in nucleus while translation on cytoplasm. Prokaryotic translation, mRNA is polycistronic, but in eukaryotic translation, mRNA is monocistronic. In prokaryotic translation, first amino acid taking part in F-met. In eukaryotic translation, first amino acid is methionine. In prokaryotic translation, it is a faster process, adds about 20 amino acids per it is a faster process, adds about 20 amino acids per second. But in eukaryotic translation, it adds 1 amino acids per second, thus a slower process. In prokaryotic translation, it requires three initiation factors, that is IF1, IF2, IF3. But in eukaryotic translation, it requires a set of 9 initiation factors. After translation, formyl group from first formulated methionine is removed, retaining methionine in the polypeptide chain. But in eukaryotic translation, the whole of initiating methionine is removed from the peptide chain. In prokaryotic translation, it requires three release factors RF1, RF2, and RF3 in the termination. In eukaryotic translation, it requires single release factor, that is ERF1. In prokaryotic translation, mRNA is short as mRNA is unstable. But in eukaryotic translation, mRNA has a life of few hours or few days. It is quite stable. Now, let's know in details about prokaryotic translation. Protein synthesis. How protein synthesis occurs in prokaryotic translation. The fundamental process of protein synthesis is the formation of peptide bond between the carboxyl group of one amino acid or at the end of a growing polypeptide chain and a free amino group on an amino acid. Proteins are made up of from a set of 20 amino acids called standard amino acids. Each of the 20 amino acids is specified by specific codons on one additional amino acid, that is selenocysteine, present in some polypeptide is directed by a modified reading of the genetic code 5' UGSA3. Polypeptide synthesis proceeds from N terminus to C terminus, and ribosome read mRNA in the 5' to 3' direction. Three kinds of RNA molecules perform different but cooperative functions in the protein synthesis. Let's know about mRNA. mRNA carries the genetic information copied from DNA in the form of series of three base codons, each of which specifies a particular amino acid. Comparison of structures of prokaryotic and eukaryotic mRNA. Eukaryotic mRNA are mostly monocistronic, having an average size of 1500 to 2000 nucleotides. It has a 5' cap which is recognized by the small ribosomal subunit. Protein synthesis therefore begins at an initiation codon near the 5' end of the mRNA. Upstream of the initiation codon contains a translatable sequences called 5' UTR or leader sequence. Similarly, non-translatable sequences at the 3' end after stop codon is termed as 3' UTR or Taylor sequence, which varies in length and sequence. In prokaryotes, most of the mRNAs are polycistronic. In contrast to eukaryotic mRNAs, the 5' end has no cap-like structure and there are multiple ribosome binding sites called Syn-Dalgarno sequences.